Hi everyone, this is the video about identifying and differentiating the nucleotides. There are two types of nucleotides. The first one is the deoxyribonucleotide. This deoxyribonucleotides is the building blocks of the DNA, while the another type is the ribonucleotide, which is the building blocks for the RNA. So both of these nucleotides, they have similar structure. Deoxyribonucleotide is having one pentose sugar connected to one nitrogenous base and one phosphate group. Same thing goes to the ribonucleotide. Ribonucleotide also having one pentose sugar, one nitrogenous base, and one phosphate group. It is the same. The only difference is their pentose sugar. Deoxyribonucleotide, their pentose sugar is deoxyribose. That means that at their carbon 2, it contains both hydrogen. They don't have the OH at carbon 2. While for ribonucleotide, their pentose sugar is the ribose sugar, which they have one H and one OH at their carbon 2. And based on their nitrogenous base, deoxyribonucleotide, they can have 4 types of nitrogenous base. It can be adenine, A. It can be thymine, T. It can be cytosine, C. And it can be guanine. There are 4 types of nitrogenous base. While for ribonucleotide, they can also have 4 types of the nitrogenous base. They can have A, adenine. They can have uracil instead of the thymine. Yeah? They will have uracil. They will have cytosine and the guanine as their nitrogenous base. So how are we going to differentiate all these type of the nucleotides with different types of the nitrogenous base? Well, actually, to differentiate it is very simple. You only need 2 simple steps. The first step is we will check on the number of the hydrogen bond. The number of the hydrogen bond that is formed in between the nitrogenous base. That's the first step. And second step would be you check on the number of the ring. The ring structure of the nitrogenous base. With these 2 simple steps, we can easily identify different types of these nucleotide, all these nucleotide. As you can see here, the first one, we can see there are 1 hydrogen bond, 2 hydrogen bond formed. Okay, so for those pair of nitrogenous base that form 2 hydrogen bond, that means that it must be A and T. It must be adenine and the thymine, because only adenine and thymine will form 2 hydrogen bond. So that is our first step. So which one would be thymine? Which one would be adenine? This one we will depends on the second step. And second step is we will check on the number of the ring. So those with 2 ring structure will be the adenine. It must be the adenine. And those with 1 ring structure must be the thymine. Then next we will look at those that can form 3 hydrogen bond. If you see 3 hydrogen bond, then that means that these must be the cytosine and the guanine. It must be C and G. So the thing is, which one is C, which one is G? So same thing, go for second step. Look at the ring structure. If you see 2 ring structure, means that this must be guanine, because only G will have 2 rings. And if let's say it's 1 ring structure, then this must be C cytosine. So with that, we can easily differentiate the ATCG based on the hydrogen bond number and also the ring number. So I have summarized all of these into the table here. So when there are two hydrogen bonds, those with one ring structure must be thymine. Those with two ring structure must be adenine. So on the other hand, when there are three hydrogen bonds formed, then those with one ring structure must be cytosine. Those with two ring structure must be the guanine. So some students might be confused. How are we going to differentiate the uracil and the thymine? Uracil and the thymine. Because if you look at uracil or thymine, it is one ring structure. But the uracil is also a one ring structure. Thymine can form two hydrogen bonds with A. Same thing goes to the uracil. Uracil can also form two hydrogen bonds as well. So their number of hydrogen bond formed and also the ring number is the same for the thymine and uracil. So how we should differentiate these two? How we can differentiate the thymine and the uracil? So the only difference that we know is that thymine is occurring, or it is a nitrogenous base of the deoxyribonucleotide, while uracil is the nitrogenous base of the ribonucleotide. That is their only difference. So based on this, then to further differentiate T and U, we will have to check on their pentose sugar. If it is a T, then at their pentose sugar, carbon 2 should not have any OH. If it is a uracil, then that means that it is ribonucleotide. Then at their carbon 2 of the ribose sugar should have the OH. So based on this, we will look at here. It is forming two hydrogen bond. It is one ring structure, meaning that it can be T or it can be U. It can be thymine, it can be uracil. So how to differentiate it? We will look at the pentose sugar here. So you can see this is the nitrogenous base. This is one pentose sugar. This is one phosphate group. So in other words, this is actually one nucleotide. So we will look at the pentose sugar of this nucleotide. So at the pentose sugar, this is oxygen. So the carbon that connect to the nitrogenous base would be the carbon number 1. This would be the carbon number 1. Then this would be the carbon number 2. The one that connected to the phosphate group, it would be the carbon number 3. Then this one would be carbon number 4. And the one that connect with the phosphate group would be carbon number 5. Okay, so we will check at their carbon number 2. We will check at the carbon number 2. So carbon number 2, is there any OH or not? We don't see any OH because they didn't draw anything. If they don't draw anything, means that this is H here and bottom plane also H also. So that's why they didn't draw it out. Okay, so you don't see any OH and they don't draw anything, means that both sides also H. So obviously when both sides also H, this is a deoxyribonucleotide. And that's why we can say that this one ring structure is a thymine. This is basically how we can differentiate the thymine and the uracil.